Now let us discuss about human reproduction. Let us see the fundamental reproduction process. We will start from the birth and we will end with the birth of the another baby. Let us take a male baby and a female baby. They grow and they reach their puberty age. After the puberty, male start producing the sperm and female starts producing ovum. Now the sperm has to meet the ovum to further proceed with the reproduction. So sperm is introduced into the female genital tract. This process is called insemination. Now in the female genital tract, the sperm travels to meet the ovum. They both meet in fallopian tube. Once they meet, they fuse. This fusion is called fertilization. Fertilization results in formation of zygote. Zygote further develops into blastocyst. Blastocyst finds a place in uterine wall to stay. So this is called implantation. Then after that, blastocyst develop into embryo and embryo development proceeds. This is called gestation. Once the embryo gets matured, it is delivered to outside world. The delivery of the embryo or fetus is called parturition. The other important thing we should remember is male produces sperm till their old age, but females they have limited number of ovums. They stop producing ovum after menopausal age. Now let us talk about male reproductive system. This system is located in pelvis. If you see the regions of pelvis, it has inside which is present in the abdominal or pelvic cavity and outside which is exposed to the external environment. This system includes pair of testes, accessory ducts, accessory glands and external genitalia. If you take testes, these pair of testes are present in a pouch. The pouch is called scrotum. Scrotum has a very important function. In our body, every enzyme works at a specific temperature, but spermatogenesis, that is, production of sperms, occur at a temperature of 2 to 2.5 degrees Celsius lower than normal body temperature. That's why the testes are placed outside the body in the scrotum. This scrotum, how it functions? When outside temperature is low, what happens? The scrotum takes testis towards the body temperature so that its temperature is maintained. If outside temperature is hot, then it takes testis away from the body. That's how scrotum maintains the temperature to produce sperms optimally. What are the shape of the testis? Testes are oval in shape. They have 4 to 5 centimeter length and 2 to 3 centimeter width. Now we study the testis from outside to inside. Outside, the testis is covered by dense coverings, and inside the testis there are 250 compartments. These are called testicular lobules. That is, each compartment is a lobule, and one lobule contains 2 to 3 seminiferous tubules. So there are 250 lobules, and each lobule has 2 to 3 seminiferous tubules. Seminiferous tubules are tube-like structure. Now inside the tube there are cells which line the tubules. There are two types of cells. One is male germ cell. Second is Sertoli cell. Among these two cells, male germ cell undergoes meiotic divisions and ultimately produce sperm. And Sertoli cells give nutrition or provide nutrition to these male germ cells, or it provides nutrition to the sperms when they are formed. Now this is structure of a seminiferous tubule, that is single tubule. Take another tubule. Now there is a space between these two tubules. That space is called interstitial space. This space is outside the tubule, which is present between the two tubules. It is called interstitial space. This space has a one important cell called Leydig cell. This Leydig cell secretes androgen hormone. The other name for Leydig cell is interstitial cell. Apart from the Leydig cell, this interstitial space also have blood vessels and other immunological cells. Now, the sperm is produced in the seminiferous tubule from the male germ cell. Now, this sperm has to come out of the body, through which all structures it travels. That we will see now. First, it enters into the reti testis. From the reti testis, it enters into the vas efferentia. From there, it enters into the epididymis, and Then it enters into the vas differentia. On the way, vas differentia is combined with another duct. The duct comes from seminal vesicle. So the vas differentia and duct from seminal vesicle join to form ejaculatory duct. And this ejaculatory duct enters the urethra. And urethra, which is starts from the bladder, the bottom of the bladder, and passes through the penis. This urethra ends as a urethral meatus. Now the sperm comes out through this urethral meatus. Again we will revise the way of sperm. It is produced in the seminiferous tubules. It enters retitestis, then vas efferentia, then epididymis, then vas differentia. Vas differentia combines with the duct of seminiferous tubule forming ejaculatory duct, and ejaculatory duct opens into the urethra, and urethra opens outside through urethral meatus. Now, you take a epididymis where it is situated. If you take a testis, it is situated on the posterior surface of the testis. And vas differentia, it ascends into the abdominal cavity and it loops around the urinary bladder. And it loops around the urinary bladder and joins the duct from seminiferous tubule to form ejaculatory duct as we discussed previously. So these retitestis, vas efferentia, vas differentia, all these are accessory ducts. Now let us see about accessory glands. There are pair of seminal vesicles which are present behind the urinary bladder, pair of bulbourethral gland which are present below the urinary bladder, and there is a prostate gland. Now these glands, seminal vesicles, bulbourethral gland and prostate gland, will have a secretions. These secretions are called seminal plasma. This seminal plasma is rich in fructose, calcium, and enzyme. When the seminal plasma combines with the sperm, it becomes semen. Now, the secretions from bulbourethral glands, apart from producing seminal plasma, the secretions of this bulbourethral glands play a role in lubrication of the penis during intercourse. Now then comes the penis. Penis, we call it as a external male genitalia or male external genitalia. It has a special tissue. That special tissue helps in erection. So erection helps in insemination. Next, it also has enlarged end. This is called glans penis, and it was covered by fold of skin, that is loose skin. That skin is called foreskin. So penis has an enlarged end which is called glans penis, which is covered by the loose skin or fold of skin, which is called foreskin. Now we will study about the female reproductive system. The female reproductive system consists of pair of ovaries, that is two ovaries, and pair of oviducts, that is two oviducts, one uterus, one cervix, one vagina, and the external genitalia. This system is located in pelvic region. These parts of the system, along with the pair of the mammary glands, that is, females have pair of mammary glands. Those parts like ovaries, oviducts, uterus, cervix, vagina, external genitalia, along with the mammary glands, they form a complete reproductive system. Ovaries, oviducts, uterus, cervix, vagina, and external genitalia, and the mammary glands, they are connected both structurally and functionally. The other term used is integrated. So why they are connected? By their connection, they help in the process of ovulation, that is release of ovum. They help in the process of fertilization, pregnancy, birth, and childcare after the birth. Childcare is nothing but breastfeeding. Now let us see about ovaries. There are two ovaries, right and left. They are present in lower abdomen. They are situated on sides. Now these ovaries are attached to the pelvic wall and the uterus by a structures called ligaments. Ligaments help in the ovaries to attach to pelvic wall and uterus. Each ovary is about 2 to 4 centimeter in length. And if you see the structure of the ovary inside, there is a ovarian stroma. This ovarian stroma is covered by the thin epithelium, and the stroma is divided into two zones. The outer zone is peripheral cortex and inner zone is inner medulla. So these ovaries are called primary female sex organs. Rest all other all secondary female sex organs. Ovaries are primary female sex organs because ovaries produce female gamete. The female gamete is ovum. And apart from the ovum, ovaries also produce steroid hormones, different types of steroid hormones. These hormones are called ovarian hormones. Fallopian tubes, also called oviducts, fallopian tubes, uterus, and vagina. All three constitute the female accessory ducts. They are together called female accessory ducts. If you see the length of the fallopian tube, it is 10 to 12 centimeter long. This length of the fallopian tube is again divided into three parts: infundibulum, ampulla, and isthmus. Fallopian tube extends from the ovary till the uterus. Infundibulum is towards the ovary and isthmus is towards the uterus. Infundibulum has a finger-like projections and they are called fimbriae. They help in the catching of ovum from the ovary. Ampulla, which is present in the mid of the fallopian tube. Ampulla is the widest part and isthmus. Isthmus is the narrowest part. Next structure is uterus. It is single. The other name for uterus is womb. The shape of the uterus is like a inverted pear. If you see a pear and invert that pear, the shape of the uterus is like that. That is inverted pear. Uterus is supported by the ligaments which are attached to the surrounding pelvic wall. Now there is a cavity inside the uterus. It is an accessory duct, no? So there is a cavity inside the uterus. This cavity opens into the vagina through a small part. That part is cervix. Okay, there is a uterus and there is a vagina below. Both are connected by cervix. The cavity inside the cervix is called cervical canal. The cervical canal and also cavity inside the vagina both forms birth canal. That is, both together is called birth canal. So birth canal consists of cervical canal and also vaginal cavity or vaginal canal. Now again come back to uterus. Uterus has three layers. Outside it is thin membrane like layer. It is called perimetrium. Then in the middle there is a thick layer which is made up of smooth muscle. It is called myometrium. What is the function of the muscle? It contracts. So this layer, the myometrium, exhibits strong contraction during delivery of the baby. The next inner layer is called endometrium. It is a glandular layer. Means this layer has a many glands. So which layer is towards the uterine cavity? Endometrium. That's why we call it lines the uterine cavity. This is the layer, the glandular layer or endometrial layer, which undergoes changes during menstruation. Means it is not constant. It becomes thickened, again thinned out. So this is cyclical change in this layer occurs during the menstruation. Female external genitalia. To understand this, let us start with vaginal opening. Above the vaginal opening, there is a urethral opening. So how many openings are present here? Two openings. Below is vaginal opening, above is urethral opening. Above the urethral opening, there is a structure called mons pubis. It is a fatty tissue. This fatty tissue is covered by skin and pubic hair. The next part is labia majora. Labia majora are said to be fleshy folds of tissue. They extend down from the mons pubis. Imagine they start from the mons pubis and surround the vaginal opening. Next, there are other pair of folds of tissue. They are called labia minora. They are just under the labia majora and they also surround the vaginal opening. Now, this labia minora join at upper end at some structure which is a tiny finger-like structure that is called clitoris. The vaginal opening is covered partially by a membrane. This membrane is called hymen. The hymen is often torn during first coitus or first intercourse. Is it always like that? No. The hymen can also be torn in sports persons who does horseback riding or cycling. So there can be a multiple reasons for the rupture of the hymen. In fact, the presence or absence of hymen is not a reliable indicator of virginity or sexual experience. Now we will see about mammary glands. Both males and females have mammary glands, but female mammary glands are functional, that is, they secrete milk. This is true in all female mammals. They are paired structures, that is, they are two in number. The mammary glands contain two kinds of tissue. One is fatty tissue and second is glandular tissue. Glandular tissue of each breast, that is one breast, is divided into 15 to 20 mammary lobes. How many lobes are there? 15 to 20 lobes. These lobes contain cluster of cells. The cluster of cells are called alveoli. And these cells, the cells which are present in the alveoli, they secrete milk. The secreted milk is stored in the cavities present in this alveoli. The alveolar cavities open into mammary tubules. The tubules of each lobe, there are many alveolar cavities in each lobe. They open into the mammary tubules. The tubules of each lobe, they join to form mammary duct. Several mammary ducts join to form a wider ampulla, which is a mammary ampulla. And this is connected to, ampulla is connected to lactiferous duct through which milk is sucked out. Two important points. In fallopian tube also, the wider region is ampulla. In mammary gland also, the wider duct is ampulla. And when baby sucks milk from the mother's breast, it is sucking from the lactiferous duct. Now we will see about gametogenesis, which are the primary sex organs in males, testes, which are the primary sex organs in females, ovaries. Testes produce sperms and ovaries produce ovum. Sperms and ovum are gametes, and the production of sperms and the ovum by the primary sex organs is called gametogenesis. Now let us see about spermatogenesis. The production of sperm by the testis is called spermatogenesis. Do you remember there are seminiferous tubules in the testis? These seminiferous tubules are lined by two types of cells, male germ cells and Sertoli cells. Now we are interested in male germ cells. They are also called as spermatogonia. Spermatogonia is a plural. That is, you tell spermatogonia when you are referring to many cells. The singular is spermatogonium. This spermatogonia, that is male germ cell, first undergo mitotic division. Remember, first undergo mitotic division. With mitotic division, the number of spermatogonia increases. Next, some of the cells of the spermatogonia, not all, some of the cells of spermatogonia undergo or get ready to undergo meiosis. The cells which are ready to undergo meiosis are called primary spermatocytes. So spermatogonia are diploid. Primary spermatocytes are also diploid. So when this primary spermatocyte undergo meiotic division, that is a first meiotic division. The first meiotic division is always reduction division. This results in the formation of two equal haploid cells. They are called secondary spermatocytes. So, secondary spermatocytes are haploid. Primary spermatocytes are diploid. Secondary spermatocytes are haploid. Now, this secondary spermatocytes undergo second meiotic division. We know that second meiotic division is same as mitotic. So, this results in the formation of four equal cells. They are haploid. These four cells are called spermatids. Now, the one primary spermatocyte is converted into four spermatids. Now, these spermatids should be transformed into sperms. The process of transformation of spermatids into the sperms is called spermiogenesis. The other name for sperms is spermatozoa. Now, after this spermiogenesis, what we have? Sperms. These sperms have heads. Now, everything is happening in seminiferous tubule where there are Sertoli cells also. So, once the sperms are formed, their heads are embedded in the Sertoli cells, and when required, they are released from the seminiferous tubules. This release of sperms from the seminiferous tubule, the process is called spermiation. See, first we have seen about spermatogenesis, next spermiogenesis, next spermiation. Total three processes we have seen here. We know that spermatogenesis starts at the age of puberty. What actually is happening during the puberty? At puberty, hypothalamus gets activated. There is a release of one hormone called gonadotropin releasing hormone, which is GnRH. This is released from hypothalamus in the brain. This gonadotropin releasing hormone acts on the anterior pituitary, releasing two hormones. One is luteinizing hormone and other is follicle stimulating hormone. Now, the luteinizing hormone and follicle stimulating hormone are released into the blood and they travel to the testis. Luteinizing hormone acts on the Leydig cells. Where are the Leydig cells are present? Between the seminiferous tubules. They act on the Leydig cells and result in the release of androgens from the Leydig cells. And these androgens act on the male germ cells or spermatogonia to start the spermatogenesis. So basically, there is a release of gonadotropin releasing hormone from the hypothalamus during the puberty down the line, which starts the spermatogenesis. Now, another hormone also released from the anterior pituitary, that is follicle stimulating hormone. What is the function of follicle stimulating hormone in males? Now, it acts in other cell of the seminiferous tubule, that is Sertoli cell, and it helps the Sertoli cell to secrete some factors which help in the process of spermiogenesis. See, luteinizing hormone is responsible for spermatogenesis. FSH through the Sertoli cells help in the process of spermiogenesis. Now, sperm is produced. Now, you want to see the shape of the sperm. You cannot see it with naked eye. To study the sperm, you should have a microscope. You should study it under microscope. So, sperm is a microscopic structure. When you see through the microscope, you will see four parts: head, neck, a middle piece, and tail. See, sperm is a cell, so it is covered by plasma membrane all throughout. That is, whole body of the sperm is covered by plasma membrane. Now, if you see a head, inside the head there is a elongated nucleus. How many chromosomes will be there in this nucleus? 23. That is haploid nucleus. Anterior portion of the nucleus is covered by some cap-like structure that is called acrosome. Zome means body, no? It is body, some body. This acrosome contains enzymes. What are the use of these enzymes? These enzymes are used during the fertilization of the ovum. We will see that soon. Next is middle piece. Basically, it is the neck, then the middle piece. Nothing much about to tell about neck. Then what does the middle piece has? The middle piece has numerous mitochondria. Mitochondria means there will be numerous ATP. Mitochondria is powerhouse, no? So, to which this power is supplied? The middle piece supply energy for the movement of the tail. So, when tail moves, the sperm is motile. That is, movement of the tail helps in the motility of the sperm. Do you remember? Once the sperm is in female genital tract, it has to travel till the fallopian tube to meet the ovum. This movement is aided by the tail motility. So that's why motility of the tail is required for fertilization. Now, during one coitus, a human male ejaculates about 200 to 300 million sperms. So, for normal fertility, to make a female pregnant, 60% of this 200 to 300 million sperms must have normal shape and size, and at least 40% of them must show vigorous motility. So, basically, normal shape, normal size, and also vigorous motility is required for successful fertilization. Next, sperms which are released from the seminiferous tubules are transported by the accessory ducts. We have seen that. During its transport through the epididymis, vas deferens, and through the urethra, the secretions from epididymis, vas deferens, seminal vesicle, and prostate are also will be there. These secretions are essential for maturation and motility of the sperms. So, see, the secretions from accessory ducts and glands are also responsible for good motility of the sperms. Now, I have told that seminal plasma about the seminal plasma. Seminal plasma is produced by the secretions from seminiferous tubules, bulbourethral glands, and prostate glands. That seminal plasma, along with the sperms, constitute the semen. So semen is a seminal plasma plus sperm. Now this male sex accessory ducts and glands, their function is maintained by testicular hormones, that is androgen. Androgens are secreted from the Leydig cells. So they are not only responsible for the spermatogenesis, they also are responsible for functions of accessory ducts and glands. Now, let us see the gametogenesis in females. Imagine you are inside the ovary of a baby, female baby, who is inside the womb. There are two kinds of cells you will see here. One is oogonia and other is mesenchymal cells. And how many of oogonia are present? There are around 2 millions of oogonia are present. Here what we have to understand is there are two important changes occurs. One is related to the oogonia and second is related to the mesenchymal cells. Let us see what happens. Again, everything is occurring in the ovary of a embryo, female embryo. Oogonia, it cannot stay, it wants to divide. So it starts its first meiotic division, but it cannot complete it. So after prophase 1, it gets arrested. Further meiotic division doesn't occur. Now this oogonia now is called primary oocyte. Now see, in mesenchymal cells, what happens? Mesenchymal cells form a layer around this primary oocyte. This is a layer of granulosa. We call it as granulosa cell layer. Now, this primary oocyte together with the granulosa cell layer is called primary follicle. Now, all 2 million primary oocytes are covered by this granulosa cell layer. So, we have around 2 million of primary follicles in the ovary. Now, female baby takes birth. From birth to puberty, no change in the primary oocyte or granulosa cell layer occurs. But one important thing occurs. Some of the primary follicles degenerate. So initially there were around 2 million. By puberty, their count comes down to 60,000 to 80,000. That is the only thing occurs from the time of birth to the puberty. Then after puberty, menstrual cycles start. Here, the development of the primary oocyte and the granulosa cell layer continues. Further layers of the cells deposit around the granulosa cell layer. That is further granulosa cell layers deposit. And also theca layer is also developed outside. Now it is called secondary follicle. What happens to the primary oocyte? Till it's like that only. Next, the cavity appears in the granulosa cell layer, which is called antrum. And the theca layer differentiate into inner internal theca or theca interna and outer theca externa. Now, this is called tertiary follicle. During the development of the tertiary follicle, there occurs something to the primary oocyte also. Primary oocyte continues its meiotic one division. So, after meiosis 1, two cells are formed. The important thing here is they are unequal cells. One is large cell, the other is small polar body. This large cell is called secondary oocyte. So we had primary oocyte. From primary oocyte, we got the secondary oocyte after finishing meiosis 1. The other side, the primary follicle developed into tertiary follicle. Now, this tertiary follicle further develops into Graafian follicle, and the secondary oocyte deposits a layer around the or builds up a layer around it. The layer or the membrane formed is called zona pellucida. One side, tertiary follicle is getting into Graafian follicle. Inside, the secondary oocyte getting a membrane called zona pellucida. Now, during ovulation, this Graafian follicle ruptures and the secondary oocyte with its zona pellucida is released outside the Graafian follicle. So, during ovulation, the female gamete is released at what stage? Secondary oocyte with its membrane zona pellucida. Now we will study about menstrual cycle. Reproductive cycle in monkeys, apes, and human beings is called menstrual cycle because here cyclical events occur. First, we will study about some terminology. A female, when she starts first menstrual cycle in her life, that period is called menarche. And at around 50 years of age, she stops menstrual cycles. That period is called menopause. So, in between the menarche and menopause, it is called reproductive phase. And each cycle is about on average of 24 to 28 days. Now, let's see what happens during this cycle. The thing is, during this cycle, there occurs changes in the release of hormones from the brain. There is changes in the ovary and also there are changes in the uterus. Let's see about one cycle. For that, now we will enter into the uterine cavity. Once you are in the uterine cavity, you see that there is a glandular tissue and many blood vessels. So that is a uterine lining. On day 1, it starts shedding off this whatever the glandular tissue and blood vessels. It gets sheds off as a liquid, and that liquid comes out of the vagina. It occurs for 3 to 4 days. This phase is called menstrual phase. The day on which the shedding of this endometrium occurs, that is the starting day of the menstrual cycle. Now, next is what happens. We will see in a sequential events. Two hormones are released, luteinizing hormone and follicle stimulating hormone. Till the 14th day, their levels gradually increases. These hormones reach the ovary. Inside the ovary, now we have primary follicles. And these hormones act on the primary follicles. The changes we have discussed previously starts occurring. That is, primary follicle is converted into Graafian follicle through the stages of secondary and tertiary follicle, and primary oocyte continues its meiotic division to convert into secondary oocyte. These changes occurs due to the under the influence of luteinizing and FSH hormones. Now, during this development of the Graafian follicle, it releases a estrogen. This estrogen acts on the uterine lining. So, with the influence of estrogen, the endometrium again grows, the blood vessels again proliferate. So that's why it is called follicular phase of menstrual cycle. Now, at around 14th day, there is a sudden increase in the secretion of LH from the pituitary luteinizing hormone. There is a sudden burst of luteinizing hormone. This sudden burst of luteinizing hormone also result in the burst of Graafian follicle. So when Graafian follicle ruptures, it releases secondary oocyte, that is ovum. We can call it as a ovulatory phase. Now this ovum reaches the fallopian tube. Here it waits for the sperm to get it fertilized. There can be two scenarios from here. Either it gets fertilized, resulting in pregnancy, or it doesn't get fertilized. First, we will see what happens when this ovum is not get fertilized. So, ovum doesn't get fertilized. Now, whatever the Graafian follicle is there, after release of the ovum, it converts into structure called corpus luteum. We have seen that developing Graafian follicle secreted estrogen. Now, this corpus luteum secretes progesterone. It secretes it for about 14 days. So this progesterone maintains that uterine lining, thinking that the fertilized ovum may come. But after 14 days or over the next 14 days, when fertilization doesn't occur, the corpus luteum degenerates. So if corpus luteum is degenerating, the progesterone levels comes down. So progesterone could not able to maintain the uterine lining. So when it degenerates completely, there is a complete absence of progesterone. Then after 14 days, there will be a again start of new menstrual phase. Suppose if pregnancy occurs, then this corpus luteum releases progesterone to maintain that uterine lining so that the zygote can be implanted in the uterine lining so that embryo can develop. So initially the progesterone is secreted by the corpus luteum. If pregnancy occurs, then the developing placenta take over and secretes the progesterone to continue the pregnancy. So we have seen four stages of menstrual cycle. The first is menstrual phase. The second is follicular phase. It is also called proliferative phase because there is a proliferation of glands and blood vessels. Then comes the luteal phase, the phase which is maintained by the corpus luteum. It is also called secretory phase. Between the follicular phase and the luteal phase, there is small phase that is the ovulation occurs, where ovulation occurs, that is ovulatory phase. Now let us see about fertilization and also implantation. During copulation, that is during coitus, semen is released from the penis into the vagina of the female. Now the semen contains sperms. These sperms are motile. They move rapidly or they swim rapidly from the vagina. They enter into the cervix, and from the cervix they enter into the uterine, and from the uterus they enter into the fallopian tube to reach the ampulla. Why it is swimming and going? It is swimming and going to meet the ovum. Now, from the ovary, the secondary oocyte or ovum is released. That also reaches the ampulla of the fallopian tube. See, if ovum has reached the ampulla and sperm has not reached, then fertilization doesn't occur. And the same way, if sperm has reached but the ovum is in that case also, fertilization doesn't occur. For fertilization to take place, both should reach there. Now, once they are in the ampulla of the fallopian tube, the sperm comes in contact with the ovum. You remember the ovum has a extra membrane, that is zona pellucida. When sperm comes in contact with the zona pellucida, remember here, semen contains millions of sperms. All will swim to the ampullary region, but the first sperm which comes in contact with the zona pellucida will change the architecture of the zona pellucida. After it gets changed, it won't allow other sperms to enter it. Once that occurs, we know that sperm has a acrosome. Acrosome has a enzymes in it. With the help of these enzymes, the sperm enter into the cytoplasm of the ovum through the zona pellucida and the plasma membrane of the ovum. There's something important occurs when sperm enters through zona pellucida and plasma membrane. This event induces the completion of the second meiotic division in the secondary oocyte. So now the secondary oocyte undergo secondary meiotic division. Here also unequal cells are formed. One is large, which is called haploid ovum, also called ooted. The other is small, which is called second polar body. Once it completes its meiotic division, the nucleus of the sperm, that is haploid nucleus of the sperm, joins the or fuses with the haploid nucleus of the ooted, forming a diploid zygote. This process is called fertilization. Now, always the haploid nucleus of the ooted have a X chromosome. If sperm containing Y chromosome fuses with the ooted, it results in the male zygote. And if sperm containing X chromosome fuses with the ooted, it results in the female zygote. So the sex of the baby is decided by the sperm, not the female. Next, now that diploid zygote is formed, it has to pass through the isthmus. From the ampulla of the fallopian tube, it passes through the isthmus to reach the uterine cavity. While moving it undergoes mitotic division. This mitotic division is called cleavage. And first it divides into 2, next 4, next 8, like that. So the 2, 4, 8, 16 daughter cell stages or these cells are called blastomeres. And the embryo with 8 and 16 cell blastomeres is called morula. Okay, now the zygote is divided into 2 cell blastomeres, next 4 cell blastomere, next 8 cell, next 16 cell, doubling. So when we call that embryo as a morula during the stages of 8 and 16 cell, then the morula also continues to divide. This division continues and it transforms into blastocyst as it moves inside the uterine cavity. This blastocyst also has a blastomeres. These blastomeres are arranged in 2 layers, the outer layer called trophoblast and the inner cell mass. The trophoblast layer is there, it gets attached to the endometrium of the uterus, and inner cell mass gets differentiated into embryo. See, the blastomeres in blastocyst arranges into 2 layers. The outer layer is called trophoblast and it attaches to the endometrium. The inner cell mass differentiate into embryo. Once it attaches, the uterine cells are there, no? The endometrial cells, they divide rapidly and covers it. Covers what? It covers the blastocyst. Now this we call embedment of the blastocyst in the endometrium of the uterus. This embedding of blastocyst in the endometrial lining is called implantation. From here, the stage of pregnancy starts. Now let us see about pregnancy and embryonic development. Now we know that the blastocyst, it is present inside the uterine lining. We also know that there is a outer layer of trophoblast cells and inner cell mass. We have to see what structures are derived from the outer trophoblastic layer and inner cell mass. The cells present in the trophoblastic layer, that is trophoblastic cell, produce a finger-like projections. These finger-like projections are called chorionic villi. The villi are surrounded by uterine tissue. See, when these cells produce finger-like projections, they project into the uterine tissue. That's why they are surrounded by uterine tissue. And this uterine tissue has a maternal blade. So this chorionic villi, together with the surrounding uterine tissue, is called placenta. It is a structural and functional unit which exists between the mother and the embryo. What is the role of placenta? It helps in transport of oxygen and nutrients from mother to embryo, and the carbon dioxide and other waste materials produced by the embryo are transported back to the mother so that mother can excrete them outside. And this placenta and embryo is connected by another structure called umbilical cord. Basically, the transport of substances occur through umbilical cord. Now, not only the placenta helps in transport of materials, it also has a endocrinology role. It secretes hormones. Hormones like human chorionic gonadotropin, human placental lactogen, estrogen and progestogens. These hormones are secreted by the placenta. There is something called relaxin, which is secreted by the ovary. Remember, this human chorionic gonadotropin, human placental lactogen, and relaxin. These three hormones are secreted in female only during pregnancy. The other hormones like estrogen, progestogen, cortisol, prolactin, and thyroxine, the level of these hormones increase to many fold in mother's plate. They are required to maintain the fetus or fetal growth, metabolic changes in the mother, and maintain the pregnancy. Now we will see what happens to the inner cell mass. Inner cell mass first differentiate into outer layer called ectoderm and inner layer called endoderm. Then after some time, the other layer called mesoderm forms in between these two layers. See, this inner cell mass has another type of special cells called stem cells. They have ability to develop into any tissue and organ. And this ectoderm, mesoderm, and endoderm are responsible for every tissue or organ in our body. Next, we will see little about the development of the embryo during the 9 months duration of pregnancy. After 1 month, the fetal heart is developed. So after 1 month of pregnancy, we can hear heart sounds. And by the end of second month, limbs and digits are formed. And by the end of third month, major organ systems are formed along with well development of limbs and digits. And by the end of 5 months, the fetus starts moves, and the movements of the fetus can be appreciated. And also there is a development of hair on the head. By the end of 6 months, small hairs appear on the body of the fetus, and eyelids starts separating, and also eyelashes also starts appearing. So like this, by the end of ninth month, the fetus is fully developed and ready to get delivered. Next, now we know that average duration of human pregnancy is about 9 months. This duration is called gestation period. After 9 months, the baby is grown to adequate size. This baby or fetus after growth, and also the placenta, which are present inside the uterus, they elicit mild uterine contractions because they want to come out. The baby want to come out. So that's why they elicit mild uterine contractions. This is called fetal ejection reflex because fetus want to eject out of the uterus. So it is called fetal ejection reflex. From this mild uterine contractions through the nerve, signals reach the pituitary. From the pituitary, oxytocin is released. Then this oxytocin goes and stimulate the uterus, leading to stronger uterine contractions. See the reflex, it started with the nerve, and the pituitary is secreting the hormone that is endocrine. So it is a neuroendocrine reflex. This reflex continues, like it stimulates the uterine contractions. Uterine contraction further leads to the more oxytocin release. This leads to the ultimately vigorous contraction of the uterus, resulting in expulsion of the baby out of the uterus through the birth canal. What is birth canal? Cervical canal plus vaginal canal is birth canal. This process of expulsion of baby out of the uterus is called parturition. Now, soon after the infant or the baby is delivered, what is left in the uterus? Placenta is left. The placenta is also expelled out of the uterus. During all this period, that is during pregnancy, the other structures are undergoing change. Those structures are mammary glands. Mammary glands of the female undergo differentiation during the pregnancy. What is the function of the mammary glands? They have to produce milk. So, after the differentiation, by the end of the pregnancy, mammary glands are ready to produce milk. And the process of producing the milk is called lactation. So once the baby is born, the lactation helps the mother in feeding the newborn. The other thing we have to know here is the milk which is produced during the initial few days of lactation is called colostrum. The milk produced during the initial few days of lactation is called colostrum. It contains several antibodies. These antibodies are essential for newborn. So the newborn won't get infections because of these antibodies. So newborns develop resistant to infection because of these antibodies. Apart from this, breastfeeding during the initial period of the infant growth is recommended to raise a healthy baby.